All right, welcome MS translators. It's my pleasure again to be joined by Dr. Travis Stiles from Novoron Bioscience in San Diego, California. Travis, thank you for joining us. We did a fantastic video a little while ago now discussing remyelination in multiple sclerosis, which if you haven't seen it yet, the link will be in the description under the video, so you can go and watch that as well. But while we talked a lot about the work that's going on into remyelination in MS, we didn't really get a chance to talk about your work in this area. And one of the reasons that we're so excited about having you as a partner on MStranslate is because you are someone who is directly working on remyelination in multiple sclerosis, which we know our community is very excited about. So for our chat today, I wanted to focus on talking about you and your work at Novoron Bioscience, because I think our community would be very interested in hearing about it. So I'm going to naively ask for a brief background on Novoron Bioscience. Can you just give us a little bit of background in terms of Novoron, where it came from and how it came to be, I guess, but before we launch into some of the MS specifics?
You know, this is probably like the funny thing is this is probably the most common thing that I'm asked to do. Like people kind of have me come in and do stuff. They're like, just tell us a little bit about your company. And it's the thing that I still like don't do very well. But you know, I'll give it my best. So Novoron actually started, the technology that our company was founded on actually came from my thesis work, which is a little bit unique. It's not very often that as a, you know, piddly little grad student, you find something that becomes your life's work. But I will spare you the narrative about the way that the discovery came up, even though I love that story. We can save that for another time. But basically the premise is I was actually in a different lab, in this lab that was primarily an inflammation cancer lab. They worked on a receptor called LRP1. And at the time, the tumor, the cancer subclass they were working on was glioblastoma. And so, you know, glioblastoma is a brain tumor. And one of the things they'd kind of like come across and serendipitously found was that this receptor, this LRP1 receptor, is actually a really robust receptor for myelin debris. And, you know, being somebody who was really interested in neurodegenerative disease and was aware especially in the spinal cord field, of how various components of degraded myelin were inhibitory to nerve repair, but not all of the machinery, not all the receptors, not all the kind of components had been identified as terms of like, they knew the myelin molecules were inhibitory, but they just they hadn't identified all the reasons why. It piqued my interest. I was actually in a seminar series when they first talked about it. And I was just like, have you guys looked at this in terms of regenerative failure? And they were like, no. And they didn't really have any interest in it. So with some persuading, I actually had to rally some of the other neuroscience faculty on campus to persuade this particular faculty member, Steve Gonius, to take the plunge and add a neuroscience arm to his very successful pathology and oncology research program. And that was it. We went in there, my first few months in the lab was just learning all the tools and kind of the getting some training from other labs around about during neuron cultures and whatnot. And, you know, we just basically had, their lab already worked with this kind of like standard lab, research reagent that acted as a receptor antagonist and plated nerves directly on purified myelin and blocked the receptor, and they grew like crazy. And that was it. That was like my first big, you know, rotation project, and it turned into my whole thesis, and now it's turned into Novoron. But just so that was the story about how the project started. But towards the end of my graduate studies, you know, we had kind of built kind of a team around this program, but like, so, you know, 3-4 years in, that some of those team members had actually started getting jobs elsewhere. So, and Alban Gaultier, who is still our biggest collaborator in the MS space, was a project scientist, and him and I worked very closely together. He taught me everything I know. But he got a faculty gig at University of Virginia at the Brain Immunology and Glia Center. And so when he left, you know, Steve, you know, to his credit, basically came to me and he's like, you know, like, once you're gone, like, we pretty much built this team and now they've all kind of moved on to other jobs. And like, so if you want to take this project with you, you're welcome, which is not super common for grad students to be said, take all the work you've done with you. Usually when you go to apply for postdocs, they want you to work on the things that their labs already work on. but he knew that the project was promising, and the university had already patented the work. They were excited about it. But as not even having a PhD yet, it was very hard for me to find a lab where I could transport projects. Like I said, like labs, when they hire a newly minted PhD to do a postdoc, they hire you to come work on stuff they're already doing. So really, Novoron started because it was the only way for us to keep the project alive. And I was too junior of a scientist to get my own lab and be able to write my own academic grants as a PI. So we created Novoron as a way to apply through the NIH, National Institute of Health, for what's called SBIR grants, which the Small Business Innovation Research Grants, which is, one of the really cool things that, the federal government here has a lot of issues, but the NIH has a lot of, is really a great group in terms of, trying to catalyze innovation. They partner with the Small Business Administration here at a certain percentage of every dollar that goes into research has to go towards translation. So not just the discoveries, which is super important, but 3 cents of every dollar has to actually go towards taking those discoveries and making them into something that's going to help people, into a product or something that's going to actually get into the patients or hospitals or whatever. And so, being able to start a company allowed me to write grants as a PI, and that's how we started. We got our very first SBIR grant in 2014. And that's what we've been doing ever since.
Okay, fantastic. So I think before we get into specifically what you're doing in the remyelination space, I think it's worth just having a quick reminder, and we've talked about this before, certainly on MStranslate, but it's worth talking about. Myelin repair isn't novel to MS. We know that it happens a lot of times in a lot of different conditions, and it just happens naturally. But that's not the case in multiple sclerosis, at least not after a period of time anyway. Whether or not it happens initially, it stops happening and there's a block. Can you just sort of, for people watching the video, explain, I guess, that concept, the fact that myelin repair does happen naturally, but for whatever reason, and we have some ideas as to what that block is, that problem, or that stops happening in MS?
Yeah. So I'm going to do what I do, and I'm going to speak in very strong terms about things that there's probably people out there that would push back and say there's more nuance to. But I think sometimes it's important to the narrative is sometimes important to know, even though there's some subtlety that might be a little bit more complex. Because like you said, in general, we think of the brain and the spinal cord as being not having a regenerative capacity. And it's not that black and white. There is some regenerative capacity. Like, we know that oftentimes, like when somebody's paralyzed in a spinal cord injury, their functional loss that they have when they present to the emergency room is not the same a level of functional loss that they're going to have in 3 months or 6 months in every year. And you kind of, in general, the rule of thumb is like, you get about 12 or 18 months before you can really say that's the best you're going to get, because you do recover some. It's just, it's not reversible. Like, it's profoundly not reversible. There's a robust deficit that remains even after the body does its very best to try to repair the damage. And in the case of spinal cord injury, a lot of that repair is the myelination, because the axon repair isn't there. Like, axons can be damaged and kind of get better, but if they're severed, if the connections are lost, they really don't do a very good job of reconnecting. And so a lot of that repair, a lot of that functional recovery that people get is because of the remyelination. And frankly, like, Novoron really started only in spinal cord injury. So you'll hear me talk a lot about this. The remyelination component of what we do actually came as a result of our spinal cord research, because the link between axon regeneration and remyelination is kind of obvious. Like, you need the electric wire and you need the plastic coating in order for things to work the way they're supposed to. And especially, I think it's important for people to know, like, most nerves have myelin on them. The myelin is really important for protecting the nerve, and also for making sure that the conduction is really efficient. And myelin in the brain and spinal cord is actually really, 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 I want to say dense, but it's just very specialized relative to like the peripheral nerves, and that makes the nerve be much more efficient. But when the myelin becomes disorganized, it's also much more complex, difficult to clear. It can persist longer and then interfere with things. But there's been people well before Novoron, well before I even joined grad school, that were looking at remyelination as a way to improve spinal cord injury outcomes. There's actually most spinal cord injury researchers will always talk about what was the big first clinical trial that everybody was really excited about. They'll talk about the Geron trial. Geron was this company where, and people will tell you, like 90% of the time, if somebody knows about this, they'll say Geron was doing a stem cell trial. They were doing a stem cell trial, and immediately what comes to mind is like people were just taking stem cells and hoping they became new nerves and replaced connections. But really, the truth is Geron was actually using an OPC progenitor cell, like an oligodendrocyte progenitor cell. So what they weren't even trying to do neurons. They were actually using a cell that they hoped would become new oligodendrocytes and help with myelin repair as a way to. And there was some evidence to suggest that maybe that would facilitate neuronal repair as well. But it's funny because that really is one of the more famous trials in spinal cord, and it really was an oligodendrocyte and therefore a myelin-based approach. And the only reason I go on that little tangent is because early on, when I joined Steve Gonius's lab and Alban and I started working together, we knew that there was, so the signaling pathways that were involved in regenerative failure for axons, there's overlap quite a bit with regenerative repression of remyelination. In particular, the same kind of molecules that will act on receptors on neurons will stop basically growth cones, which is like, you have a wire and you cut it. What happens is the little wire will try to regroup and form a little hand to try to crawl forward and find a new place to connect. But when that little growth cone hits myelin debris, it gets confused and just stops. When those same molecules interact with oligodendrocyte progenitor cells, so taking a step back, oligodendrocytes are the cells that myelinate your axons. They're very sensitive cells because one oligodendrocyte can literally wrap around and myelinate hundreds of axons. So when you mess with them, they really don't have a chance to recover. They just die. And then there's this pool of progenitor oligodendrocytes, basically baby immature oligodendrocytes, that will come in and clean up the mess. And once the mess is cleaned up, they'll mature and they'll wrap and repair the damage. But when there's myelin debris around, those progenitor cells, they'll still come in and they'll try to clean up the mess. But they do a lot of the early job really well, but they stop short of actually remyelinating. They don't actually end up sending out projections and wrapping around axons again. And so we always knew that was kind of because those events kind of overlapped, that there was a potential for a remyelination approach to what we were doing. And when I was in grad school, Alban worked a lot on that side, on the glia side, while I worked on the neuron side. But it wasn't until we actually started doing animal studies in spinal cord injured animals that we started seeing that, on the order of days, like if we would infuse an LRP1 antagonist into the spinal cord after a spinal cord injury, that's not enough time. Days or even a week or two isn't enough time to actually see an axon regrow. It takes quite a few days for the axon to even kind of regroup so they can form that growth cone. I would say it's probably 2 weeks before it can actually start trying to mount a forward moving response to try to regenerate itself. But we saw in days myelin changes. So not only did we see that we were able to kind of encourage more cells to start trying to look like they're myelinating, but we actually kind of spared or protected some of the oligodendrocytes that would normally kind of probably die from secondary damage to kind of stabilize them. And that was when we knew, okay, there was always this idea that there might be something here, but if we didn't expect to see a robust myelin phenotype in our spinal cord injury models. And when we did, we really were like, okay, we need to have a remyelination program because this is actually even more robust than some of the axon things we were seeing. But all of that is not related to your specific question of talking about, well, why do we get some remyelination in some indications and not in MS? And the truth is if you get a concussion, if you get even some viruses will cause demyelination. And for the very kind of majority of the time, it's you can actually have a pretty comprehensive recovery. There are a lot of theories about why that doesn't happen in MS, but the truth is it's not even the same in every MS patient. There are some MS patients that can have RRMS who the first few attacks they have, is if you were able to control their disease and kind of limit future attacks, they might actually recover almost all the way. At least it like for the most part, it would functionally appear that they'd actually regained most of the function, even though they probably haven't. There's probably still some deficit. But there's something that, so one of the factors that is different in MS is the repetitiveness, the repeat, insults to different regions of the brain. And that can lead to kind of like, I would call it a fibrotic response. I don't know a lot of people that would call it that. I think that's one of the things that overlaps in a lot of these is kind of like various aspects of like fibrosis, but you get scarring. And, if you can't do really much to the body where you traumatize a small section of, an area of the body over and over again without getting some sort of scarring or calcification. The brain's no different, right? The other side of it is if it's sometimes people get really, really robust attacks. And so not only do you have this this myelin debris that's in a much larger region, so it's harder to clean up, but you also have this immune infiltration in this really intense inflammatory environment, which also complicates things. And that doesn't really happen as much like in like a traumatic brain injury, where, it's really the bleeding and swelling. But once that kind of resolves, there's not as much of an immune activation because it's not an antigen recognition. Like there's a difference between like the body recognizing there's a wound versus the body thinking that it needs to attack something. And that's what happens in MS. The body actually attacks the myelin. So not only are you bringing soldiers to the battlefront, but they think that that's the enemy, and they get much more fired up, and that creates a much more kind of detrimental environment. But it's none of, like I said, none of it's really binary. Like the repetitiveness of it is a problem. The severity of the inflammation, the type of the inflammation that comes from the kind of the autoimmune trigger, all those things really exacerbate an already suspect, not super efficient system. And that makes it all much, much harder. So that was a really long answer, and I'm not even sure I answered your questions completely, but hopefully there was some useful stuff in there.
Yeah, no, I think that makes a lot of sense. And so, I think there's something to be said, and this is a question that I'm going to ask you a bit later in terms of timeframes for these sort of things. But when I've been talking about this recently to people and trying to explain why I am hopeful about why we will see a remyelination therapy, is because we know there's a block, but people are looking at multiple ways around that block. And I've been using an analogy which I'm not 100% sure that I love, but I got positive feedback on it the last time I used it. So I'm going to roll with it for a little while. Of if you're trying to go to the shops and you're driving along a road, and then on one day you come across a tree that's fallen down across the road and you can't get past it. There are a number of different things that you could do in that situation. You can try and remove the tree from the road so that you can just go about life as normal. You can remove that block. Or you could find a new way of getting to the shops, find a completely different path to get around it, or you could somehow jump over that tree and start it from there. And to me, I think that's how we're looking at remyelination therapies. We're either trying to remove the block that's causing the problem. And one of those things you already talk about is, can we clear myelin debris? Because we know that that's an issue. Or if we know where the block exists in these pathways, can we just somehow jumpstart that pathway after that block that exists? Or can we find another way around it? And so I think, for me, that's what makes me hopeful that we're looking at all of these different things. But I guess talking about Novoron specifically, how are you, as much as you can say, because we know that there's obviously a lot of stuff that can't necessarily always talk about completely at different stages, but how is what you're looking into remyelination? How does it avoid that block?
So I have very strong opinions on this, because I don't think a lot of people are going to agree with me, but because I think most people, if you ask them what the blocks are, they would give you biology, some sort of medical issue that is, it's the myelin debris, it's the scarring, it's the inflammation, it's the blood-brain barrier, it's, whatever, T cells, macrophage dysfunction, microglia hyperactivation, all those things are real. If I'm being really honest with you, though, if you had to ask me if there's one thing that's stopping us from actually getting a remyelination drug into the clinic, I would say it has nothing to do with biology. It's science. I think that we don't do enough to make sure that the way we're using a drug or a therapy or a treatment or even a device is the most efficient. I think that when it comes to things like remyelination, there is no path. Nobody's been pioneered to say this is how it's done. Nobody's been successful. So nobody really knows the best way to go about it. And so a lot of times we build off iterative findings where a positive finding is considered good enough to keep moving. And so people will get good enough data, and they'll iterate and they'll iterate. But if you're not optimizing, if you're not maximizing how well the drug's working or how well the therapy's working, and you're just operating under the assumption that the fact that it's working is good enough to move to the next step, if you do that enough times, you then eventually get to the place where you're getting 10%, 20% of the actual benefit of your drug. And that's in a disease that's as heterogeneous and complicated as MS, that might not pan out to show benefit in a clinical trial. I think that one of the things that I actually am very passionate about with Novoron that I think makes us different, we've talked about this in the past, is that I am focused first and foremost on giving the drug the very best chance to succeed and working backwards. I'm not interested in limiting what I think we can do by saying it's too invasive or there's too many risks or et cetera, et cetera. Like, I want to know what it takes to get the drug to work, and then we can work backwards from there to make it more convenient or simple or palatable to a patient or whatnot. When we went to New Zealand, I gave the tea kettle example, where when I asked all the patients in the room, if I could cure your MS, but it required me to pour the drug directly into an eye socket and take your eyeball out and pour it directly into the eye socket, that cured your MS, would you do it? And, everybody in the room, after being disgusted by my horrible analogy, they eventually raised their hands, because obviously they don't want this disease, and they would sacrifice a lot to fix it. But when you talk to drug developers, especially in pharma, there's a risk aversion where no, there's no way we would do something that aggressive with putting a port in a head or, putting a needle through the skull or, things like that are far less invasive that, if it would work, would you do it? Their answer was no. And we've had conversations where it's like a clear no. If we had to reinvent the wheel and find a new way to inject into the brain or put a pump into the skull or something, whatever crazy thing you can come up with, there's not a really strong kind of appetite to go after it. Not to say that it doesn't exist, but it's not a common approach. So that's what I'm the most focused on. I actually start, like, one of my fundamental assumptions about this field is that I believe our approach can work. I want to make it to the best approach possible. But one of my baseline assumptions is, like, I don't think we're the first remyelination drug that can actually improve my remyelination. I actually think, and somebody's going to watch this and eventually get mad at me for saying this, but I actually think that the Biogen anti-lingo antibody it worked. Like, they showed that in their acute optic neuritis patients that they improved nerve conduction velocity, which it's very hard to say that wasn't because they actually improved remyelination, but the drug is dead because they took a really blunt force approach to have their clinical trial in the larger MS population and their RMS patients. And there's many flaws in that clinical trial design that I think killed the drug. So it wasn't that it didn't work. It was just that they weren't strategic enough in their trial design. Now I'm going to get in trouble for saying that. And so I take a lot of pride in, like, not repeating the same mistakes, and making sure that if our drug works, it's not because we go about testing it the wrong way that makes it fail. Like, when we get there, if our drug is either is once we get to a clinical trial, my number one goal is to make sure that when we get there, that we will feel as confident as we possibly can that a positive result is because the drug works, and a negative result is because it doesn't, and there's no other ancillary factors that can get in the way. So I know there's much more fun answers about science that I think people would probably be excited to hear about, but I think it's important to emphasize that in this space, there's kind of more simple and practical issues in play that we can address that aren't always emphasized enough.
Well, and I think it's important for people to realize as well that it's a very different space as a biotech company developing this research in-house to a large pharmaceutical company, in that you really only have one shot at this.
Oh yeah, for sure.
And not to lump the pressure on you with this sort of stuff, but the nature of this research is that these things aren't cheap. It's difficult to get funding, and so you can't afford to make mistakes. And I know we've talked a lot at conferences we've attended about how strongly you feel that, not from a company perspective necessarily, but from a community perspective, that what you really struggle with is the idea that something that may work doesn't get pursued not because the drug isn't effective, but because there's mistakes along the way. And the reality is that you can't write in the space that you're in. You can't go back and just go, okay, let's do this again and try and change things and make it work. You just won't have that opportunity. And I don't think people realize that.
I like you said it very well. I think that there's a moral responsibility here to not risk falling short, because it's not just us. I mean, Biogen is a billions and billions of dollar company, and they did get more than one shot. They got like a 1.5 shots, but I can guarantee you that the scientific team behind that program is not happy that program got pulled. And it got pulled because shareholders were not happy with the failure of that clinical trial, and there was enough momentum to squash it. And so if it can happen to Biogen, we have no shot. If they can have a program shut down that they believe in, and I know that they believe in it, and they're good, and there are well-intentioned people there that had very good ideas and plans for what they wanted to do. I don't think that they appreciated that a failure in the RRMS trial was going to stigmatize the whole program and make it difficult for them to move forward. I think I can tell you that they were very excited about the optic neuritis trial. They thought that was going to be an iterative step where they could work towards getting broader and broader approvals for their drug. But the lack of patience in wanting to rush forward in the RRMS space, because it was the bigger market, caused them to want to go that route. And they made the compromise. They did both trials. And I'm not going to sit here and pretend like I don't understand that logic. Like, it seems like a good compromise. Okay, fine, we'll do both. That's a big commitment on the part of the company. That's a big financial commitment to, to this drug that you can't pretend is sinister or a lack of commitment. That's a big commitment. It's just, I don't think anybody was prepared for, if the optic neuritis trial works, but the RRMS doesn't. I don't think anybody was prepared for the RRMS trial to completely overshadow the success of the optic neuritis trial. I still am kind of befuddled by that. The optic neuritis trial was, a first ever in human proof of concept that we can remyelinate axons. That's insane. That's amazing. That's a revolutionary. And people don't talk about that because, opicinumab is now stigmatized as a failed drug, and it's unfortunate.
Yeah, and I mean, I think it's important at this point, and we'll move away from the anti-lingo discussion in a second, because I want to focus more on your work. But it's important to realize, and you said it there, that this wasn't it wasn't a necessarily bad thing on the part of Biogen. These trial designs are difficult. It's very easy. There are many questions, and it's hard to get right. And I think that's where we're going to go through a really long-term build up to this, and not saying that they didn't do that, but to try and give ourselves as much information as we can before we go into that space. And I think we were all excited about that and all disappointed when it didn't work. But I think it just also highlights how tough these things are and how careful we need to be in the planning that's there. And so, when we talk a little bit later about timeframes for this, as much as we all understand, I mean, we don't completely understand, because neither you or I are living with multiple sclerosis, but we understand that there is people want this now, and we wish we could give it now, but there are reasons why we need to take a little bit more time with this than we would like, and not just get everything into clinical trials, because that's how things fail, and that's how we don't end up with something when we wanted to.
Well, and I think that's important to point out, because it's easy to pick on Biogen now, because you can look back and you can have the benefit of hindsight to pick holes in what they did. But they committed a lot of money to trying to do things, and they included a lot of patients. There is a case to be made for them actually being more dedicated to the cause than a lot of pharmaceutical companies would be. they just made a mistake, and a mistake you can learn from.
It's tough to be the first. I mean, it's tough to be one of the first people doing these large clinical trials. And the reality is, while it's disappointing that they failed, everyone else has learned from that. We get learnings from things that don't work, and the important thing is that we don't repeat those mistakes. And I think that's what we're going through. So in terms of Novoron's MS program, can you just give us I was going to say brief again, but I'm not even going to bother. Can you just
I'm sorry.
Can you just tell us a little bit about where that program is at the moment?
Sure. So I'll be very honest with you. Like when we first got that spinal cord data, the first thing we really wanted to do is to take our time and make sure, can we translate this into MS models? And so before trying to repurpose our existing kind of lead molecule for spinal cord injury as an MS drug, we actually did the tedious work of going through and creating like selective transgenic animals to see if we could confirm, like validate LRP1 as a therapeutic target for remyelination. So we did both. We created OPC specific LRP knockouts, because it's the OPCs that are actually interacting with these myelin molecules and then failing to differentiate.
Those are oligodendrocyte precursor cells that we talked about before.
Yeah, the baby oligodendrocytes, the baby myelinating cells. And interestingly, though, as a side note, one of the things that was a really cool clue to us when we were starting this is there is a ton of LRP1 on the OPCs, the baby oligodendrocytes, like one of the if not the highest expressing LRP cells in the brain. Oligodendrocytes that are mature, almost no LRP1. So it's actually interesting. So like one of the things you worry about when you do genetic knockouts as a proof of concept to validate a therapeutic target, you worry about, well, what other stuff is going to go wrong when we knock out this entire gene, right? But it actually just works out really well that mature oligodendrocytes apparently don't need LRP1 to do their job. So it was actually a really serendipitous kind of model. Because we weren't going to be affecting the actual myelination itself. But the results we got were really, kind of blew our minds. We actually had problems early on because we thought we weren't in the EAE groups. We thought we were doing something wrong with the immunizations, because most of those animals will eventually get sick enough where you have to put them down because they're just too ill and you don't want them to suffer. And not enough animals, because it was a blinded study, right? Because not enough animals were getting sick, and we thought something was wrong with our immunizations. And we were actually playing with getting new adjuvants so that we could try to make the animals sicker, because they weren't getting sick enough. And it wasn't until we kind of unblinded things that we realized that the reason that we didn't think they were getting sick enough is because almost none of the knockout animals were actually showing symptoms. And that was just like the first kind of like light bulb, like, holy cow, like this could be really cool. And then we did a second model with a Cooper zone model where it's one of the most boring kind of pieces of data that we show people, but it was actually one of the ones to me that made me feel really good about what we were doing being the right path. Because, there's a couple things that happen when oligodendrocytes are killed and the oligodendrocyte progenitors kind of come in to try to replace them. So you'll get cell migration. So all these baby oligodendrocytes, OPCs, will start swimming into the region, right? So you'll see a bunch of them in an immature state. Then once they get there, they actually kind of work as like, kind of like construction workers for a little bit, cleaning up the mess and making sure everything's nice and tidy. And then once they're done cleaning up the mess, then they start to differentiate. But the first thing they do, because they're kind of normal blobby cells in the beginning, but oligodendrocytes, mature oligodendrocytes, are like octopuses with 100 arms that wrap around 100 different axons, right? And so that's a lot of cell membrane. It's a lot of like surface area. So the first thing they do before they start spitting out projections is they swell up to make their membrane expand so they can have enough membrane to send out little projections. And so in our first study, there's markers of immature oligodendrocytes and mature oligodendrocytes. And we were just looking at the ratio of immature to mature as kind of our endpoint. Like that's kind of like the standard of what's done. We hadn't really thought about doing it in either way. That's usually good enough. And the ratio changed. It was really good. It was a really robust result. But one of the things that we saw that we didn't expect to see, we weren't even really looking for, is the size of the cells. You did not need to measure these things. You looked at them and you're like, those cells are massive. They look like little chubby, obese jelly bean babies. And like, it was just really obvious that even though it was an unanticipated finding, the fact that we were seeing this like really, really significant change in the hypertrophy of the cells, it was like, man, this is a thing. This is something, right? Unanticipated findings in science are always some of the best evidence of what you're doing. Like, because for better or worse, like if you don't expect to see something, you can't be biased by it. You can't bias yourself in terms of an expected result. So a lot of times when your unexpected findings are supportive of your your primary results you're looking for, it's comforting. Alternatively, if you have unexpected findings that run contrary to your hypothesis, you should really start to reevaluate things, because those are often more telling than your actual primary things you're looking for. Yeah, so that was a big part of what we did. I'm sorry, again, super long tangent there. But so our lead molecule is actually Novo-118. Novo-118 is what we want to use for remyelination. And this is going to be a little bit. This is important, so it's a little longer, but it's going to be. I apologize in advance if people tune out during this part. But so Novo-118 works really well in cell culture. Beautiful. In an animal model where we can just inject a bunch of it into the brain, boom, great, awesome. The problem is Novo-118 is a comprehensive antagonist of LRP1. So it's like napalm for your weed problem in your garden. It's a bit excessive. And so in terms of like being able to use it over long periods of time, it's potentially suboptimal. If you could have a more surgical approach, that would actually be preferable. Now, for spinal cord injury, you don't need to be surgical. It's an immediate injury. It happened one time. You need to treat for a few weeks, and then you never have to worry about it again. So if it's aggressive, that's fine, because once you stop putting the drug in there, whatever went wrong is not happening anymore, and you're fine. Now, theoretically, like one of the other things that we're working on is an Alzheimer's program. Now, we wouldn't be able to do this for Alzheimer's, because there's a difference between having to give an animal a drug for weeks versus decades. Like, whatever bad things might happen with an excessive approach, a scorched earth approach is not going to be sustainable if you're doing that for years and years and years. Now, MS, in theory, like we think Novo-118, so we do not think Novo-118 is the ideal candidate for an Alzheimer's program. We do think it's perfect, and it's actually a beautiful thing for spinal cord. MS is kind of in the middle, because we're not, and this is the difference between us and what Biogen did with ofaginumab, because Biogen thought that this was going to be a drug that they had patients on forever. And I think that was fundamentally one of the biggest mistakes they made, because remyelination, you have a window for remyelination. Once a lesion is resolved and whatever extracellular matrix has been deposited, you're not coaxing more myelin into that space, not without doing something else first to get rid of all the scarring, the fibrosis, right? So there's a bit of an overlap with a newly formed or an active MS lesion and a spinal cord lesion. Just the sheer nature of the fact that we refer to them both as lesions indicates to you that it's an acute situation. So we don't have to treat them forever. That's it. And I think that that's a big distinction, is like, thinking you're going to have patients on this drug for the rest of their life every day is, one, not necessary. And two, it gives us this ability to be a little bit more aggressive. So there's kind of two arms here that we're looking at. So we're not taking it for granted that Novo-118 is going to be the right drug for MS, because even though we can do an acute treatment, there still is going to be the need for multiple treatments through a patient's life. And so we're not under the illusion that if we stop an attack or reverse the damage from an attack, that's going to stop them from having more attacks later. And what we don't want to do is have a drug that, if over repeat doses, might cause some issues. We don't know. We have reasons to think that that's not going to be a huge problem, but it is worth considering that that's not ideal. So on one side, we have platforms that we're working on, so that we can try to make a more surgical approach, so that we can specifically target the LRP domains that are responsible for remyelination failure, and leave the rest of it intact. One of the things about LRP1 that makes it really unique to almost any other drug target is it's about 10 times bigger than most other receptors. It's like 600 kilodaltons, which is just how we measure protein sizes. And, most things are between 25 and maybe 100 at most. And a lot of times, though, in those drugs or in those molecules, we actually know the very specific place we want to target. LRP, we don't. It's massive, and it has it's not like that big chunk of it that's floating around outside the cell has only one specific kind of like binding domain. It has a lot. And so being able to know which specific one is important for the remyelination phenotype and targeting that specifically, that's one arm of what we're doing. We're seeing if we can hone our approach to be more precise for the remyelination phenotype so that we can eliminate some of the off-target effects. On the other side, we're still moving forward with Novo 118. And on that side, the hard work that we're doing is focusing on what is the best way to deliver the drug. So like like I mentioned before, like we can inject it into the brain and we can get benefits, but can we do IV? Can we do subcutaneous? Can we put in a catheter and infuse it? And then, one of the things that gets tedious, but is really important work is figuring out what the protocol is going to be for how you use a drug. How often are you going to give it? At what dose? At what frequency? What flow rate? Like all these different things. So that, so we're trying to make sure that we move on both fronts. So that we can try to give Novo 118 the best shot. But if that ends up looking like it might not be the best long-term solution for MS patients, we're also working on the next generation of drug that can be a little bit more precise. Was that ridiculously long?
No, I think it makes a lot of sense. And so I mean, a couple of comments on that. I think in terms of we don't necessarily need this to be something that's given every day for the rest of a person's life. I think we're probably in a position now as well with the developments of other treatments that aren't remyelination therapies, but they're, immunomodulatory, immunosuppressive therapies. Now we're at a point where some of those are really effective. And so hopefully that is slowing down the new damage to a point where we don't need to be able to, we're not giving a remyelination therapy all of the time, but they can be given, as you say, little by little. So I think that, the timing of that is even better because of how other things have developed. And I think in terms of how you talk about Novo 118 and this, it's similar to what we heard around the bexarotene trial from the team at the University of Cambridge. And we'll drop the link to that interview in here as well, where they did a clinical trial with something that was more that, as you describe it, napalm approach, which they knew that their drug was quite messy, and they knew that it wasn't necessarily what they wanted. It wasn't that surgical approach that you talk about as well. But because that bexarotene was already an approved drug, it was easier for them to trial that. And they found that dirty approach, while it was effective and they could see remyelination, had way too many side effects associated with it. And so that won't be pursued. And so I think, what you talk about in terms of how Novoron's approaching this has a lot of grounding in terms of what we've seen elsewhere. So I think that's, great. Last question, I guess, around timings of things. And I'm going to ask you this in two parts, one regarding your work and one just in general, because that's the question that I get all the time, and you're probably even better to comment on it than me. But first question around Novoron's work. Do you have timeframes for when you think this may get to clinical trials? And the short answer to that may be just no, that's too hard to predict, and that's fine. Second question around that is, in your expert opinion, being someone who's working in the field, what do you think is a realistic timeframe? I guess one, before we start to see sort of large clinical trials for remyelination therapy, so those sort of phase 3 trials. And also, if all goes well, when we might start to see something on the market for that.
So I'll answer the first question. So in terms of when I think we'll hit the market, if you'd asked me that question a year and a half ago, I would have given you a much different answer because I was extremely cavalier about what I thought we would do. I think in the best scenario, I think we're looking at 3.5 years before. No, that's not true. In the best case scenario, because it's the same drug as spinal cord injury, assuming it would be Novo 118, we could be looking at as soon as like 3 years, right? Being in the clinic, in clinical trials. And because it's an orphan condition, it can move pretty quick. You usually want to give, as a general, really rough rule of thumb, you want to say like 8 years to get through clinical trial. But I think a good, robust drug for remyelination, you could cut that in half in theory. Now, the reason I don't want to like commit to that time frame is because, like I said, what if we have a better way? We might only get one shot on goal here. So like if Novo 118 is 75% as good as what we think is 2 years behind, we'll probably punt and go 2 years and wait 2 years for the better drug. Only because the worst thing we can possibly do is stigmatize a program that has the potential to work. I know that there are patients out there that are desperate, and it keeps me up at night, the fact that a delay means that there are patients that might pass away that don't get a chance to try a drug. And that sucks. Like, that's the part where you really start to feel like, there's a paradox here. But I really feel like, that window, that couple year window of time where you might lose some people, is a necessary sacrifice for the perpetual benefit you'll have if you actually get something approved. And that's just that's one side of this. In terms of like, when I think we might see like a good phase 3 clinical trial for remyelination, I will say this, Biogen did a lot well with what they did. I think Teva's actually done some cool things too. I think AbbVie's doing some really cool things. It's easy for me to poke holes in what other people do, but I think, Biogen took a big shot, and they put all their, they went all in, and unfortunately they had a couple big critical mistakes, but man, you got to give them a ton of credit for the swing they took. There's a lot of companies that would have done less. Teva, and I believe Teva, like when they bought the Remyelination Foundation and took on some of that technology, that was a big swing on their part. I think that there's going to be stuff coming along. I think that it's possible. I hope, and this is, my investors would be mad at me for saying this, but like, I hope somebody beats us to it. I really do. One, I think there's going to be room on the market for multiple approaches. I don't think anybody else is doing what we're doing. So in theory, if somebody comes at it from a lingo approach or a small molecule approach or whatever, those things might be synergistic, and that would be amazing to kind of like, piggyback on something and make an even better, get better outcomes. But I think that there's now, I didn't think this was true, and I don't think it was true in the years past, but I do think that people now are starting to learn lessons and saying like, look, if we're going to do this, let's make sure it's the best version of what we want to do. And part of the reason I feel that way is because whenever I see a company that is looking at remyelination, and then a year, 2 years, 3 years down the road, they're like, oh, by the way, now we're thinking about also doing a spinal cord program. To me, that's a really good sign, because to me, it means that they were seeing what you would expect to see from something that actually works. It's not intuitive that you would link a remyelination approach to a spinal cord approach. But when people start to naturally come to those conclusions and realize that their drug could have those benefits in that system as well, you start to really think, they might be on something, because that's what happened to us. Like we weren't necessarily expecting to see the robust response that we did, but there it was. And so I do think that there's a couple companies out there that might be a little bit under the radar, especially because they're a little bit smaller, that are going to be popping up in the next 12 months, and that are going to be something to be excited about. And I say that because I think before the kind of second half of the pandemic, there wasn't a ton of investment money interested in spinal cord injury, remyelination, maybe a little bit more, but still not nearly as much as cancer or diabetes or some of these other things. But the second half of the pandemic, for some reason, the number of dollars that went into startup biotech went through the roof. And so I think a lot of these companies are finally capitalized, and you won't see the results of that yet because they're probably still ramping up and getting some proof of concept. But I think, I'm hopeful that over the next 12 months that there's a lot of companies like ours that are even probably better funded than us, that are going to start coming out with some cool stuff. So I think that there's reason to be hopeful. There's a handful of large pharma approaches that I think will be interesting to watch as well. But I just think that there's some really kind of outside the box thinking out there in some of these in the startup world, that people that are onto something are gonna start making waves. So obviously, like, I don't want to be too naive about the mountain we have to climb, but I think we really are getting close.
Yeah, I mean, I think one thing to emphasize at this point, and we did it with our other video that we talked about is that in these discussions, what we hope the MStranslate community likes is that you and I are taking the liberty here to chat and to be very honest and just to share our thoughts. And a lot of the things that we're sharing, I mean, we're very clear when we're talking about data and studies, if we're talking about that versus these are our opinions. And I think people appreciate that, you know, we're sharing our opinions and our thoughts on this. And so, you know, we're going to be right sometimes, we're going to be wrong other times.
Yeah.
Hindsight is always 2020, and forward thinking couldn't be any blurrier, but, you know, we're trying to share our thoughts on that.
Well, and I gotta be honest, like, I love these conversations because we can be honest, because we are very candid about the things we say. And I can say a lot of the things that there are environments where I can't speak this candidly because you never know who you're going to ruffle feathers with. Like someday there's some of these videos someday are going to really, like, people are going to be like, I saw you on MStranslate, don't talk to me. And I'm going to be like, oh, that's too bad. But sure here's the thing. They might not say it out loud, but I think it's really important. You and I have talked about this a lot. I think it's really important. Patients and patient advocates can do a lot to move the needle in terms of what we emphasize and how we do things. And like, you know, I think one of the worst things that can happen are when people lead really motivated people down the wrong paths, and they can convince them, you know, stem cells is notorious for this. Like, you know, everybody gets excited about stem cells because you have people that are better at PR than science, and people start saying, we need more stem cell research, and they put all this energy into it. And some of the better science doesn't really get as much attention. And so that can steer things, that can steer momentum, enthusiasm, it can steer funding, it can, you know, progress. And I think the conversations that we have like this, that are when, you know, some of those really, you know, great motivated people, you know, can see a more nuanced conversation, it gives them the tools where I think that they can, you know, take that energy and advocate for much more positive things. And I think you and I have always been, you know, from the first time that we had chatted about this stuff, we were very much aligned on that like nothing gets better if the patient community isn't as informed as they can be. And so what point is there in sugarcoating or biting my tongue? Like, people don't have to take my word for it. I'm going to be wrong about a lot of this stuff, but I certainly want them to have a candid perspective from somebody who at least thinks about these things every day. You know what I mean? As a starting point.
Yeah, no, I completely agree. And so, you know, as we always say for people watching these videos, if you do like them, if you don't like them, whatever it is, please do just engage with them. You know, click the like button, click the share button. If you want other people to be able to see them, make sure we get as many eyes on these as video, and comment below. Let us know what you think about them. Give us feedback, ask questions, and we'll make sure that we respond to all of those questions. I'm going to finish up with I'm going to get a single word answer from you on this, just a yes or no. I'm holding you to it. A yes or no.
I'm sorry, have we met?
Yeah.
I don't know.
All right, people at this point are 40 to 50 minutes into the video. So we're going to keep this one to
the one that wasn't this supposed to be 15 to 20 minutes?
I think I said that before we started recording. Yeah. So I've learned my lesson. When people ask me realistically, when might we see remyelination therapy? I say that I'm hopeful that we might see something within the next 5 to 10 years. Do you agree with that? Yes or no?
Yes.
Okay, there we go. So if you made it to this part of the video, there's that short answer for you. Travis, thank you as always so much for your time. These conversations are great. We were talking before this that we may try and do regular video podcast style things where Travis and I just chat. We have a lot of things that we like to talk about. We enjoy talking, as you've certainly picked up from this video. So again, if you have topics that you'd like us to discuss in those, drop them in the comment section. We'll make sure we include those as part of our future videos. Travis, thank you so much. We're excited to continue to hear more about the fantastic work that you're doing, and also just to get you involved more and more talking to our MStranslate community.
Thank you. Always a pleasure.